I'm going to really give you a brief introduction and leave enough time for my friends and colleagues who are going to be the stars of the show. And I'll also introduce them all at the end so that we don't have to have lengthy introductions throughout each of the talks. This is actually a very nasty disease and something that I think many of you are familiar with. We've talked about it a lot. And unfortunately, if you compare pancreas cancer to the other common and uncommon cancers, there's been very little tangible progress in terms of meaningful 5-year survival, which is still about just 10 or 11%. It's gone up by about 1% a decade, which is really disappointing. What we feel and what we're going to try to show you is that some of the research that we're doing and other work around the world that with the other groups that we're collaborating with should make a significant impact over the next little while. Just for those who aren't that familiar with the anatomy, the pancreas is an organ in the retroperitoneum, meaning it's in the back. It's sort of behind the peritoneal cavity. It's kind of a silent spot, which is one of the reasons why the disease is so nasty, is that the tumors develop in many cases silently. The tumors in the tail here and the body are often quite silent until they cause pain, and by that point, they're often metastasized. Tumors that occur here in the head, these patients are in quotes lucky because it blocks the bile duct and they can turn jaundiced, and those are the patients that seem to have a little more favorable outcome. The other anatomic features is that some major arteries and veins, particularly the celiac artery, the SMA, the portal vein, and other mesenteric veins travel right through the head of the pancreas through the Uncinate process, and these tumors often become invasive and technically more difficult. The good news over the past 30 years is that we've kind of maxed out on surgical safety. These are some seminal trials from the early, well, about 20 years ago, where what are called volume outcome studies. It's sort of the practice makes perfect philosophy where groups or hospitals, more so the hospital than the surgeons, who do high volume work, meaning 10 or 20 or 50 cases, which are on the right, have better outcomes when you look at survival. And in Ontario, we actually, because we have a single payer system, we can control or regionalize healthcare a little bit more easily than in the US and in other countries. So a panel was convened in the early 2000s to come up with standards for pancreas cancer surgery, which traditionally was actually quite dangerous. And in many cases in the 70s and 80s, the outcomes from surgery, the complications and deaths from the operation were higher than the cure rates from the cancers. So we've actually made surgery quite safe now in Ontario. There's only about 10 or 12 hospitals that are allowed to do the work. And these centers all meet certain criteria, minimum of 2 surgeons, 24/7 interventional radiology. These are many doctors that help us with some of the complications. We have fellows full-time, nurses who are highly trained. And as a result, surgery is safer. But despite that, this kind of heroic surgery that we do at UHN, this can take 10 or 12 hours where the tumor can be wrapped around some of these vessels. Vessels have to be reconstructed. But even in those cases, sometimes we wonder if it's even worth it. The outcomes are not that good. Patients are very desperate and they'll sign up for these operations. But we have to be very careful that we're not overdoing it in a disease that's often systemic in nature. Only about 15 or 20% of patients have curable or potentially curable resectable tumors. The other disappointing feature that we're going to address today is systemic therapy, meaning chemotherapy. And this is sort of a timeline of 3-year survival for over the past many decades. Drugs such as gemcitabine were brought in place in the 80s. Now we have a cocktail called FOLFIRINOX. But despite that, we have less than 1 year survival for patients who are receiving the most toxic or most modern chemotherapy. And we really need to make significant progress. The other problem is that pharma has been fairly nihilistic in addressing pancreas cancer. There's much more interest today, but we still see some very disappointing trials, even just recently, of novel agents that just didn't work. And in some cases, because the phase 2 data were either overestimated and the results did not pan out in phase 3, but probably more importantly, and I think Jennifer and others will address it, is that we need to recognize that there are unique subtypes of the disease. And if you develop a biologic agent for a specific subtype, you should probably only be studying that subtype in your randomized trials. And this is a notion that pharma is sort of starting to wrap their heads around. The other misconception probably started around this time, and this was a paper that came out from another group at Hopkins, suggesting that the development of the disease from some sort of precursor to resectable disease to advanced disease could take about 13 or 15 years. And this opened up people's eyes and wondering if there's an opportunity for screening. I'm not going to talk about it now, but I can talk about it in the discussion. The problem with this model is it didn't fit clinically. We just don't see patients with small early lesions that are curable very often. And in fact, we see patients with small tumors. This tumor here is only about 1 centimeter in size. But in most cases, even though these are resectable patients, they often recur and die within a couple of years of surgery. So these are some of the really important questions that we're addressing in PanCuRx and some of the other collaborative work that we're doing. The other unique aspect of pancreas cancer that's been highlighted by many groups is the desmoplastic or intense tumor microenvironment with a lot of fibrosis. The group here over the past, I guess, almost 10 years now has taken the bold approach where we actually won't just take the blob of tumor, but we do laser capture microdissection. And this is because of the unique relationship we have with terrific pathologists and the groups here in enriching our samples for our genomic research that Faiyaz is going to talk about. The other important work that was done actually by Faiyaz when he was a postdoc here was studying the genomic instability that's associated with pancreas cancer, primarily chromothripsis. And that model that I just showed you from Hopkins, where the sequence of events could take 5 or 10 or 15 years, probably just isn't true, and it doesn't fit the clinical pattern. And it looks more likely that it's an accelerated growth. And this is, again, something that we need to study scientifically before we can translate that into proper screening techniques. So before I introduce my next speakers, I just want to recognize the fact that we also appreciate that our patients need to be more actively involved in our research, and we've just started the OICR, PDAC, PFAC, a lot of letters, but this represents the patient advisory committee that we've just opened up. Libby Znaimer and Pierre Herto are leading it, and Julie's helping us recruit other family members. So I won't belabor all the introductions. I think many of you know these people. They're all my friends and colleagues, so it's hard to speak about them in this sort of semi-professional way, even though they're all terrific professionals, but they're more so my friends than scientists in my mind. Faiyaz received his PhD here with John Dick, and then he worked with us early on as his postdoc with Tom Hudson and myself. And much of the work that we've done over the past 6 or 7 years in genomics has really been on the heels of his hard work, and the battery is running low. The battery's running low. And I won't, hopefully we can rescue the battery. Irene is actually a pathology resident who we recruited into Faiyaz's lab just a few years ago. Maybe she'll give the timeline, seems like forever, but she's going to finish soon and return back to her pathology residency. She's been doing some terrific work on organoids that she'll talk to you about as well. And of course, Jennifer, who's kind of my partner in crime at Princess Margaret. We co-lead the McCain Center for Pancreas Cancer, which is a very large program, which has worked really closely with the OICR as well. So without taking too much of Faiyaz's time, Faiyaz.
Thanks, Steve, and thanks to Shola for organizing, and thanks to Julie for helping out the slides. So I've been at OICR since 2011. So I've seen this program go from essentially, nothing to, what it is today. So I thought I'd do something a little bit different and give you, the journey of how we established the program. And there's been a lot of turnover, or even, a lot of new faces here at OICR in the past 5 years. And I believe was one of the flagship programs here. And so this is how we all started. Okay, so let's start off with the pancreas. So the pancreas really has two main functions. It's called an exocrine function. So the majority of its mass is dedicated to a network of ductal cells. No, go ahead, Sylvia. A network of ductal cells that basically end in these flowery organs called the acinar cells. And this is what makes all your enzymes to digest food. And there's 2% of its mass is dedicated to what's called the endocrine function. And the endocrine function serves very important homeostatic functions for controlling blood sugar. So the history of the pancreas, I feel like, is really interesting. So it was discovered more than 2,000 years ago. So if you translate pancreas from Greek, or it's Greek Latin, the translation means all flesh. And if you've ever seen the pancreas, it's a really fleshy, flimsy organ. The main Pancreas is kind of defined by this main duct, and this main duct is called the duct of Wirsung. And the main duct was actually discovered in the 1600s, but what's really interesting about this story is that Johann Wirsung was actually killed over the discovery by his graduate student. And there's a controversy over who discovered the main duct, and actually it was a graduate student that should have deserved all the credit, and this is why we give students and trainees first authorship now on all papers, because we're really scared. So in the 1800s, Paul Langerhans actually discovered the islets, and the islets are where your beta cells are located that secrete insulin. Probably, I think what some of the most seminal work in history of the pancreas came in the late 1800s by two physicians, Minkowski and Marin. So they removed the pancreas from dogs, and then they made observation. The observation was that when they urinated, there was flies attracted to that urine. And the reason the flies were attracted to the urine is because it contained sugar, and when they analyzed it, they saw sugar in there. And that was actually the seminal work that established that something in the pancreas actually controls blood sugar. And that actually set off the race for insulin and the discovery by Benting and Best in 1921, which is either in this building or the building across the street. I forgot that discovered insulin. Okay, so that's your brief intro to the pancreas history. So when we say pancreatic cancer, so pancreatic ductal adenocarcinoma is the most common form of pancreatic cancer. When most people are referring to pancreatic cancer, this is what they're referring to. And part of the reason, as Steve has introduced it, it's so aggressive is that around 80% of patients present with what is called advanced disease. 50% are, metastatic, which means that the disease is already spread. 30% of patients are locally advanced, which means that, the disease is kind of wrapped around some blood vessels and it's very difficult to remove. And only 20% of patients actually qualify for surgery. And in a lot of other cancers that these numbers tend to be flipped. So as Steve has introduced, why do we study pancreatic cancer here at OICR? Well, it's probably one of the worst cancers that exists. There's been very little improvement in survival despite advances in cancer biology and therapy over the last few decades. There are a lot of challenges to studying this disease. It will be the second leading cause of cancer-related death, despite being a rare cancer in the next decade. The survival is now reaching around 10%, which means that 90% of patients diagnosed with this disease will die within 5 years of being diagnosed, most within the first year or two. So, the risk factors have been well identified, but unfortunately, it's not like, tobacco or UV, where there's a clear etiology. And that makes primary and secondary prevention quite difficult to do in this disease. This disease spreads rapidly, it's drug resistant, trials that fail, they continue to fail. Most advances in this disease have been related to advances in how chemotherapy is used. Broad systemic therapies like immunotherapy are not working. And, as we said, most patients present with advanced disease. And so there's quite a few challenges with biospecimen collection. And so, a lot of research up until our program was created was focused on, studying only early stage disease. And this still happens all around the world. Late 2000s to early 2000s, when genome sequencing kind of took off, the US and the UK and big groups around the world were saying, we're just going to study all cancers separately, and there's going to be a lot of doubling of efforts at that time. And so Tom Hudson, who was the previous president of OICR, came up with the idea that, instead of doubling all the world's efforts, we're going to, make a consortium, the International Genome Cancer Consortium. And it was actually hosted here at OICR since 2008. So although the host was at OICR, Toronto took on the sequencing effort for pancreatic cancer alongside with colleagues from Australia. If you've ever looked at our data on the cluster here, most of the IDs are like PCSI, and that stems from Pancreatic Cancer Sequencing Initiative, which is the initial ICGC program launched in 2008 for pancreatic cancer. So the initial program was called PanCuRx. There was 3 different ideas. There are 3 different stages. Of course, we wanted to study early detection. Most of the work related to early stage disease, finding new prognostic biomarkers or biomarkers for adjuvant chemotherapy. And of course, we want to translate that in the advanced setting. And so if you wonder why it's called PanCuRx, actually, PanCuRx was trademarked in 2013, so we couldn't call it PanCuRx. Julie knows the story better, but I think it was someone from drug discovery that said, it's better called PanCuRx. RX sounds like prescription. And if you say it really fast, it sounds like pancreas. And so we called it PanCuRx. Internally, a lot of us that were still here still refer to it as PanCure. So all of the sequencing effort were wrapped around like 6 different projects. And there were 6 different projects. So molecular classification of this disease, initially, there was a lot of exome sequencing done, and we were going to produce half, and Australia was going to produce half, and we wanted to find all these recurrent mutations that are being discovered in all other cancers. And we're going to discover these mutations and pass them on to drug discovery to discover new molecules to treat this disease. Of course, there's going to be network and pathway analysis, finding new biomarkers and new biological networks in this disease. For detection purposes, the program launched to develop like more high resolution imaging to see this cancer earlier. At that time, Toronto was a hotspot for hypoxia research and pancreatic cancer. There was a program launched around that, and I joined as a postdoc in studying tumor stroma, although I did nothing to do with stroma, but that's another story. I think, I just put this up there. We had this really good program initially launched, but the problem, in hindsight was that, there were no targetable driver mutations that were discovered from the sequencing initiatives initially, unlike the scenario in a lot of other cancers. And so we had no new targets to pass on to anything else. And so the coding sequence we had analyzed at that time, we're like, okay, we need to move to whole genomes and see if there's anything else outside of the coding region. But the problem is that exomes you can sequence deep, but genomes you can't sequence deep. And the cellularity issue was a big concern. And so we couldn't sequence whole genomes deeply. And so this is like a histology slide that Steve had also presented. I think I have the same one, which is that if you take a pancreatic cancer mass, only like 10 to 20% of the mass is actually cancer cells. And so it's really difficult to analyze this on a whole genome sequencing level, only at 30x resolution or 40x resolution. We had an initial review in 2012, external review. All these really big, important scientists came to review us, and the review went quite poorly because we obviously couldn't find anything with all the sequencing effort. And what had happened is that the external review board actually had suggested that all of the projects you have to come under a specialist for pancreatic cancer. And this is when Steve actually joined the program. So Steve joined the program in 2013 as the lead of the Pancreatic Cancer Sequencing Initiative. And the other more important person that joined at that time to get us organized was Julie. And so Julie joined at the same time and got us all organized. We had a mini retreat, and we kind of changed the mission of the program. And the mission of the program turned a lot more translational at that time. And so that was one of the first important changes to the program. One of the big headaches that Steve had to conquer initially was that all of the trial workflow had to change, and we had to insert this part early on in the tumor processing. You can't see it, but you can see the green highlighted part here, which is that we had to incorporate UHN Biobank and Pathology here, that any biospecimen coming through the pipeline for sequencing had to be laser capture microdissected. And that for us was a big game changer. So I used to analyze a lot of copy number structural variants at the time. So you can see on the left here that there is basically no changes in the tumor. You can't see them, but with laser capture microdissection, you can clearly tell the amplification. The structural variant jumps by 3-fold. And then this is actually one of the, this allowed us to kind of like see the genome at a lot more detail and study the mutational process that guide this disease. And so this was one of the first landmark papers that we had in the program, and studying structural variation from whole genomes that had been laser capture microdissected. And I won't bore you with the details of all this reconstruction of structural variation. But you found that a lot of drivers in this disease were kind of altered altogether. And this kind of changed the model of how we view. So, and this has been published for a long time. So pancreatic cancer is always depicted as this, gradually growing tumor that always goes through these series of precursors. There's a large window of opportunity for early detection, and that doesn't jive with what we see at the whole genome level. At that time, Ashton Connor, who was a surgical resident, had decided he also wanted to do his PhD. He'd also published some really important papers for the program. And, we went from a program that was publishing nothing to publishing in Nature and Cancer Cell and JAMA Oncology. And all of this initial work was essentially done from early stage disease and resectable. And there was a large, I wouldn't say uproar, but there was a push that we needed to focus more on advanced disease. And so we kind of turned face at that time. Steve got in contact with Jennifer and brought Jennifer on board. So Jennifer's a medical oncologist, GI medical oncologist at Princess Margaret. And with Jennifer, we launched the COMPASS trial, and this was the first trial of its kind, focused only on advanced pancreatic cancer, biopsy, percutaneous biopsy with laser capture microdissection, focused only on metastatic or advanced disease, mostly metastatic. And it's still the only trial of its kind in the world when it comes to genomics of advanced disease. And this was the first paper published at that time for the COMPASS trial. And what the COMPASS trial did actually for us is that it turned another phase of the disease for us. So we went from just studying genomes to studying the transcriptome. There's a lot of stuff with molecular subtyping in this disease. When I had first joined the program, at that time, no one really thought there were molecular subtypes of pancreatic cancer. And it's because this disease is so aggressive. Everyone thought that the disease was uniform. The drivers are essentially uniform in everyone. Why would there be heterogeneity? And so with some changes in the past, 5 or 10 years in this idea, we started studying molecular subtypes of this disease. We focused on advanced disease. And what we found is that, there's 2 different molecular subtypes, basal-like and classical, and they actually have predictive and prognostic value. And this could not have been done without the COMPASS trial. So this was initial work from the COMPASS trial. We accrued a few more samples, and we found that basal and classical actually dramatically underestimate the degree of heterogeneity, in this disease. Initially, you could see there's a big gradient of gene expression, which makes molecular subtyping of any cancer quite difficult. And we doubled the number, and this is the work of Ganho Zhang here. And there's 5 distinct molecular blocks of this disease, and they have predictive prognostic value. And this is the work we're kind of doing right now internally, and I'm sure you're going to hear about it in the next year or two with different people presenting at the OICR seminars. So we went from a program to that essentially just was local. We focused on maybe a couple of collaborations to collaborating, internationally. Our data sets are used internationally, and this is important. And the reason it's important is because, without pancreatic cancer is a big problem. And if we don't collaborate with these major institutes and do what we do, it's never going to get solved. I put this slide again because it really does take a village to do anything that we do. There's minimum of 40 people involved, from getting consents to biopsies, radiology, all of that before you even get the sample at OICR for sequencing. And that's thanks to all of our clinical collaborations that we have now. So we went from sequencing to running genomics-based clinical trials. First was COMPASS, another, PASS1 is being run right now, another 2 are coming. And beyond just the clinical trials, we do a lot of, I think, great research here. There's detection research related to plasma whole genome sequencing, RNA sequencing, studies of the tumor microenvironment, radiomics. And so, Sola told us to take away one message. So I put this message, hopefully it resonates. So I felt like it was a few of us at the time. We had this big dream, and we started small, but I think the most important part is that we just kept persisting. And it's thanks actually to the seed funding provided by OICR to continue the Pancreas program, and that allowed us to establish what we have today. So we have a lot of people to thank. I apologize for the people that have I've forgotten on this slide, but there's a lot more than just this. And here's our acknowledgements. And I'll pass it on to Irene. Irene is one of our extremely bright graduate students that has established a really important platform for us here that I think is going to make a big difference in the coming years with respect to organoids and high-throughput drug screening.
Thanks, Faiyaz. I'm a pathology resident, and I actually graduated medical school in 2017, which, as my cohort reminds me, is a very long time ago. But I joined the PanCuRx program in 2019. I actually first encountered Steve as an intern on his surgical HPV service. and recall spending a month there, mostly just seeing people dying, which really motivated me to start working more in the molecular aspects of cancer. And so I joined PanCuRx in 2019, and I'll definitely say that I've benefited quite a bit from the structure here at OICR and also the many clinical trials and translational aspects of the research. So as we know, pancreatic cancer doesn't have a lot of systemic therapy, and because surgery is not an option for most patients, having good chemotherapy and systemic options is really important for these patients. Although there has been some progress, most of the progress has been in standard chemotherapy. And so unlike in other cancers, there hasn't been as many targeted treatments that have come out for pancreatic cancer, with the exception of, can you see this mouse here? Yeah, with the exception of PARP inhibitors for germline BRCA cases. And so part of why this is because in the past, for the screening trials that have been done, most of them have actually been done without any pancreatic cancer in them at all. Pancreatic cancer has traditionally been very difficult to grow in vitro. And so, for example, a lot of the progress that we've seen in melanoma, lung, colon, breast, is because of these 60 cell lines, which included a lot of those types of cancers. And so even in more sort of modern cell line screening efforts, the number of pancreatic cancers total is still fairly small. And in organoid studies, which have now been able to grow a bit more pancreas, what we find is mostly that either they have a lot of samples, but only kind of standard of care drugs, or they try a lot of new drugs, but they don't have a lot of samples. And so the one noticeable example that is different is in AML, which is a cancer that tends to expand fairly well ex vivo. And so they were able to do this with many drugs and many patients in the ex vivo setting. And so in one of these AML-directed trials, they were able to do the screening. And crucially, when they found different alterations that were either sensitive to drugs in specific patients, they were able to invent new clinical trials to actually test these genome drug associations. And crucially, of these patients that were enrolled in a Beat AML sub-trial versus ones that were not, their overall survival approximately doubled for those that were matched to therapy. So in the pancreas cohort, what we've tried to do then is to develop this high-throughput organoid drug screening program. And I had the fortune to start in 2019 as the COMPASS trial was kind of wrapping up. And the Living Biobank had helped us produce a lot of organoids as a pilot for the COMPASS trial. And so with the proof of concept that organoids could be grown from at least half of people with pancreatic cancer, we started expanding them and obtaining them for more different patients in order to perform high throughput drug screening in a very high throughput format, with drugs enriched for various diverse mechanisms of action in a way that we could not only look at which drugs were active in pancreatic cancer, but also which ones were more active in some pancreatic cancers than others. And so not only to see which ones were hits, but which ones specifically could target certain patients in a way that might be more targeted and less cytotoxic. Not all PDOs grow from people. And one important population that I'd just like to draw attention to are the people who do have BRCA mutations, or homologous recombination deficiency. And so interestingly, we have more success growing organoids from those that are actually HR proficient. And in general, the organoids do seem to represent a bit more aggressive tumors in general. For example, patients that tend to survive fewer days also tend to establish more organoids. But overall, our success rate has improved a bit over the years from about 40% to now closer to 70%, which has helped us to screen more patients as well and lead us more towards a potential clinically tractable assay in the future. So what everybody wants to know is, do these organoids actually recapitulate the patients in vitro? Do the drug responses that grow out actually reflect what's going to happen in the patients? And so what we find is that in the FOLFIRINOX setting from the COMPASS trial, we were able to produce 22 matched organoids with patients that had also received FOLFIRINOX, and the concordance rate was about 70%. So it was pretty good, but it wasn't perfect. But one thing that I'd like to just draw your attention to, so on top here we have the patient data. So the RESIST, basically these are patients that didn't respond well to chemotherapy, and these are ones that did. And then so in the organoids here, you can see that the red ones are the ones that were more resistant to chemotherapy compared to the whole cohort versus the ones that were more sensitive. And so there's more red on this side and more blue on this side. But crucially, we have certain cases where the organoid response shows something different than what would necessarily be predicted genomically. For example, this organoid is germline BRCA mutated and was HR deficient. And so this patient really should have had a good response to platinum, as we've seen in clinical trials. But actually, this patient progressed after only 1 or 2 cycles of FOLFIRINOX and rapidly died. And accordingly, the organoid was also similarly resistant to all of these drugs. And so this is where we're hoping that some of the functional aspects of the organoid can help pick apart differential responses, even in cases where genomic markers may or may not be there. So we developed this high-throughput screening assay, which is able to screen these organoids in a very miniaturized format and with about 3,000 drugs at once. And so the high-throughput assay is pretty reproducible biologically. So if you screen the same organoid at 2 different passages, the correlation is quite good. If you look at various different organoids across 2 drugs that are in the same pathway, the correlation is also quite good. You can see the organoids here at what we would call 100% versus 10% viability. And we've been doing this long enough now that we've actually gone through a couple different iterations of the same drug libraries. And you can see that the correlation is slightly less good than the biological replicates, but this is about in line with other large-scale screening studies like PRISM and such. And then crucially, when we go from the high-throughput screening assay with the 3,000 drugs to validation dose response curves, our correlation is even better, as you can see here. So we're aiming for 100, and we're not quite there yet, but we're almost there. And so this is we're up to 83 organoids now, and so you can see from the functional landscape that there's quite a variety of drug sensitivity across pancreatic cancer. So I'm just showing you here the drugs that have induced sensitivity in at least one organoid. And we divide these sensitivities into now what we've called, these have misaligned a bit, but you can see here what we call private hits, selective hits, and public hits. And so traditionally, most drug screening assays would focus on what we call these public hits that indiscriminately kill most of the organoids, because these are the ones that we think, oh, you can apply these to most of the patients. But the issue is that most of these drugs tend to be the ones that also end up being quite toxic in clinical trials, like proteasome inhibitors, or HDAC inhibitors or traditional chemotherapy. And where we've decided to focus with some interest are these what we call the selective and the more private drugs, which seem to impact many organoids, but not some of them, indicating that they may be responding to some sort of specific either genomic or transcriptomic lesion within the tumor. And so you can see that these are rather specific to most organoids after we do the dose response curve screening. And crucially, every organoid has some kind of unique sensitivity. So it's not just that some organoids are more sensitive and some are just completely resistant. It's the idea that every single organoid has a slightly different sensitivity profile based off of their secondary alterations. And so from some of this functional data, we can look at kind of these different networks of drugs and connect them not only by their annotated functions, but also by their pure functional results. This graph was made with the help of a very talented undergraduate student, Berkay. And so from this, we're starting to look at some of the different groups of drugs that may cluster together in ways that are unpredictable. And so, for example, some of the ones that we do predict are the ones that have known genome drug association. So those that are p53 wild type tend to have sensitivity to these MDM2 inhibitors, which only work on p53 wild type organoids, as you can see here. So this helps tell us that the assay is capturing associations of interest. But then from those clusters, we actually find that there are some novel associations which have not been previously known before. So for example, we originally only had one organoid here, and now we've got the second one of these 2 unusual male patients who have drug sensitivity to these sort of essentially female estrogenic hormones, as well as this unusual antiplatelet drug called anagrelide. And so this was recently reported a couple of years ago as related to this biomarker PD3A, which was not known to be anti-cancer or anti-tumor in any way. And then later was shown to have an interaction with this other sort of unknown family of proteins called the schlafins. And accordingly, one of our organoids, which we had RNA data on, was an outlier in PD3A expression. And so we have put this in xenograft models, and accordingly, when you treat these mice with subcutaneous tumors with anagrelide, you get a very dramatic reduction in tumor size, even after some amount of tumor growth And you can see here that in the control, this is essentially all tumor, whereas after treatment with anagrelide, we actually had a near pathologic CR after only 2 weeks of treatment. And so you can just see one lone tumor gland here. So does this actually work in patients is the next big question. And so we did have one patient who had a very early recurrence after pancreatic cancer surgery. And so we had originally developed her organoid from her surgical resection and found that she was very sensitive to the standard of care drugs, irinotecan or oxaliplatin. And accordingly, this matched her initial response. So she was treated with FOLFIRINOX adjuvantly after surgery and had a very good response to FOLFIRINOX. Unfortunately, she had a second liver recurrence after that, and the drug screen from her original organoid had also showed sensitivity to many TEC1/WEE1 inhibitors and cell cycling pathways. Unfortunately, we were unable to obtain any of these drugs for her, but we did have access to a drug in a similar pathway that was in clinical trials at Princess Margaret. And so interestingly, she actually did manage to have a partial response to the trial drug in the same pathway with a tumor reduction of about -29%. She had a progression-free survival of about 4 months and actually still remains alive after about 11 months. You can see the reduction in the tumor size here. And she had this unusual genome with CCNE1 amplification and this tandem duplicator phenotype, which is somewhat similar to the HRD phenotype, but also different in many ways. And so these are the kinds of genomic, phenotypic, functional correlations that we're trying to discover with the organoids. So the next step is this trial that we will be running with the clinical team called ADOPT. So with the Prosper Panc, which is kind of the next phase of PanCuRx, which will be this patient tissue sampling along with organoid growth and drug screening throughout Ontario. We've also obtained funding to do a small amount of patient treatment with either currently Health Canada approved drugs or with drugs that are available through the current trials pipeline. And also pursuing partnerships with other pharmaceutical companies. And so with these, we will try to match a couple 25 patients with interesting and achievable drug selections that are safe to these drugs in order to see if organoid matching is actually able to improve outcomes in the second line setting. And so with that, I'd like to thank the many people involved in this work, especially our technician, Jamay, who has helped with a lot of the high-throughput part of the drug screen, and also special thanks to Rob Grant, one of the oncologists who has done a lot of work on ADOPT.
Good afternoon. It's a real pleasure to be here. I'm Jennifer Knox, and I'm going to now turn a little bit towards using genomic sequencing to guide treatment for pancreatic cancer. And I always like to add one case at a time because it still feels like that. But briefly, I'm going to show you a little bit about the COMPASS, the PASS1, and Prosper Panc. So as the previous speakers have pointed out, you know this, pancreas has moved into the second leading cause of death, and it certainly is a strong outlier compared to other cancers, although I think we flipped the double digits now for 5-year survival. And as Steve has explained, there's a lot of reasons for this. They're late to present, they're aggressive systemically, and we have to be honest, our treatments that we use are relatively ineffective, and that includes surgery, chemotherapy, radiation, and immunotherapy. And as Steve alluded to, we do a lot of very large randomized trials in pancreas cancer, and we report a lot of very negative trials, and we haven't been able to get survival past the 1-year mark. So in the clinical world, we have two good, well, relatively good cocktails, FOLFIRINOX and gemcitabine nab-paclitaxel. Both of these have moved the needle for pancreas cancer. They have improved survival over earlier treatments, but the truth is they're toxic, and we don't know how to use them properly in that we have a conversation and we let the patient guide us based on the side effects, on the schedule. And in reality, what we really want is to be able to tell them this one's going to work for you, this one isn't going to work for you, so let's not waste time, precious time on ineffective therapies. So really what we're talking about is precision medicine, which is where patients are matched to treatments based on biological characteristics of their tumor or perhaps a clinical parameter. So the right treatment, the right person at the right time. So basically the science informs the targeted approach. This is an extremely appealing idea to patients and clinicians for obvious answers, because it sounds like it's going to be a great leap forward moving the needle. And so it was really that background that we decided, for many reasons, to take a deep dive into the genomics of metastatic pancreas cancer, which really is COMPASS. So COMPASS started back in 2015, and we decided that we were really going to power it on feasibility, because it was not easy to do at that time, to prospectively identify subgroups of patients with advanced pancreas cancer who might have distinct genomic characteristics that might then inform better treatment. And so really it was big biopsies at the beginning to look at DNA, RNA, and then PDOs added in. And they got the standard of care, but we followed them along to see what could happen. And this is this powerful database that we now have of clinically annotated genomic data. So this is the very first case of pancreas biopsy on December, right before Christmas, 2015. And this picture just highlights Sandra Fisher, the pathologist, because it was very key to have this connection between the clinical and the lab, which could not have happened without her. So over time, we expanded out across other sites like GTA and other parts in Canada. We looked at over 300 patients. We got whole genomes and RNA. And I think we certainly did learn a lot about molecular subtypes. But the main observation we had early was this idea that there are different RNA signatures, and they may predict for response to chemotherapy, which is incredibly important if it really pans out to be true. So here's our very early paper that showed that the basals were all growing on chemotherapy, and the purple ones, the classics, were tending to have a better response to chemotherapy. So that's a powerful message we're still trying to prove. So from that, we looked at a lot of different things, including, as Faiyaz mentioned, trying to see if the transcription factor GATA6 could distinguish classical and basal much easier than sequencing, other ways of looking at subtypes, and then, of course, digging in deep with organoids, not just with Irene and Faiyaz's group, but also with the Tuvison lab in the US, to really see if these are good models to recapitulate early on in a patient who has a short survival, whether they can be useful in treating them. We also looked at HANT, the nuclear transporter in COMPASS, to show that it predicted for response to nab-paclitaxel. More work on subtypes, including mismatch repair, which is a small group, but you shouldn't miss them, and understanding HRD in pancreas a lot better. And even looking very elegantly at the TME and seeing subsets within the TME from the COMPASS data. So we learned an awful lot from COMPASS, but I would just like to focus that one of the key lessons was feasibility is absolutely everything. And we've learned that not everybody can do this, and it's a lot of hard work. And others have pointed out the team that's involved. And feasibility isn't just important for the specimens, but also for patients, because the very best clinical care is when clinical care interacts with research care, and back again is the best outcomes. So this graph shows fairly modestly that we did impact survival in those patients in the red that could get a matched treatment that we found on COMPASS compared to those that could not. Right patient, right time, right drug. But you have to be honest, if we look at the numbers that we enrolled and the numbers that we could match, the intention to treat was still well less than 10%. Now, we shouldn't be too hard on ourselves, because if you look at the big US study, the Know Your Tumor initiative that looked at next generation sequencing, they also showed that if you could drug a target, you had a better survival than if you had the target and you didn't get the drug in the turquoise versus people without targets. So nice proof of principle. But again, if you look at the numbers of those that were enrolled, those that actually had tumor, those that actually could get DNA, those that could actually get a sequence and then move towards a targeted approach, intention to treat there was only 2.6%. So we've got a long way to go to improve on this. So this brings us to this timeline that when we sort of finished up learning from COMPASS, we devised a number of prospective trials where we thought we could start to impact on patient care more than just observing genomes. So the first one I'm going to talk about today is PASS1, which is a randomized trial where we're looking at genomic plus PDOs, North American wide. And the other one is this Prosper Panc, which Irene has mentioned, where we're doing the same thing, but more focused in Ontario with a little deeper science added in as well. And the point of these arrows here are really to show our strategy for some time now has been to try to slowly increase the precision choices for patients, but also the number of patients in Ontario that we're actually impacting. So here's the schema for PASS1 you've probably seen before. This is first-line metastatic disease, and they get randomized between the two standards of care, which is ethical. They've never been compared. And the primary endpoint is progression-free survival. But they get these big baseline biopsies for genomes and PDOs and also blood biopsies, so that we have this ability to look at more about chemosensitivity signatures, molecular analysis, and PDO pharmacotyping in a prospective trial on a randomized backbone. And then what gets quite exciting is our molecular tumor boards, where patients are discussed before their first 8-week scan. So the first look at how they're doing on chemotherapy. We review the cases, we look at the DNA, we look at the PDOs, what's available. We do a lit review of anything that is relevant, and then we have a sort of fulsome discussion, and we make some recommendations that the treating physician can use or discuss with their patient or ignore. But the idea really is to try to move these towards better decisions rather than empirically. And this is accrual. You can't run a trial without people asking about accrual. So here we are in purple. We're running a little bit behind what we had proposed, but there's been a pandemic and a North American wide Abraxane shortage. So all things considered, we've done amazing. And, here is a sort of an oncoprint of some of the really interesting findings that we see. Each column is a patient. And, I think you could agree that there's a lot of heterogeneity here. So for example, TMB, we found a couple of patients that almost had like a microsatellite high signal, even though they didn't have it in their germline. You can see that most of the patients are KRAS wild type, but we're starting to understand the imbalances of KRAS mutations as well as the different alleles, because those are coming along with drugs in the near future. You can see this group here that are KRAS wild type, but all of them seem to have an alteration in BRAF, which is highly druggable, and those patients get drugs and they do much better. So this is kind of exciting. This is the high throughput PDO screen down in Cold Spring Harbor, where they grow the PDOs. They're evaluating a smaller group, sort of the standard drugs plus the ones we have in trials, sequencing them and then plotting these ways of comparing the relative resistance versus sensitivity and feeding them back to us. So there's a lot of excitement ongoing here with exploratory analysis. And of course, we're batching some immunoprofiling and some circulating tumor DNA that still needs to be looked at, but there's just tremendous potential coming out of this. But again, as Irene said, what we really want to know is, do these really predict how patients will do, and can we use them on the next study to choose who goes on what? So I just wanted to take a minute to explain a case, because there's nothing like a case to really exemplify the kind of information that we get out of a patient on PASS. So this is a 66-year-old man who presented with metastatic pancreas cancer with a lot of pain and weight loss, no family history, poorly differentiated adenocarcinoma. He was randomized to the gem-nab-paclitaxel arm. Thankfully, had a deep partial response. 58% shrinkage at 8 weeks is unusually good for pancreas cancer, and he felt a lot better, and it's ongoing, thankfully. But what's really interesting is that the molecular tumor board It turns out he has a BRAF S&D fusion, which are pretty rare in pancreas cancer, but not so rare in the acinar histology. In addition, he had an HRD detect score, which was very high, really suggesting that he was a HRD case of unknown etiology, and we found no genes in his panel. So the pathologist revised based on the genomic data that this is probably an acinar adenocarcinoma mixed. The PDO is a bit slow to grow, but it's probably pending at this point. And while he's doing well on first line, the good news is he's got options and the doctor has time to get them ready for him. So you could target the BRAF with MEK inhibitors, which we know are active, or you could target the HRD signal with platinum, PARP, and DDR agents. So that's kind of exciting. So if you look at past, this is very much a work in progress and it may change. But if you look at the patients enrolled, we've enrolled 93 out of 150 at this point. About 60% of pancreas patients, this is pretty good for this trial, go on to second line therapies. And if you look here in this turquoise, some of these decisions are very empiric, whatever is available for the patient, versus these other ones that are correlate driven, either based on the genomic findings or on the PDO findings. So roughly at this point, almost 63% of patients are having a genomic or a correlate based decision, which doesn't necessarily prove they're living longer, but it certainly proves that our processes and our communication is working. And with more data, we'll be able to show this. So I find that really exciting. So I'm going to stop because PASS is a work in progress and spend the rest of the time on Prosper Panc, which I've alluded to. So basically, this is a platform in which patients can come on this a state of the science, look at their tumor at any place in their journey, whether it's localized disease or advanced. We're picking a slightly younger population because we want to have a better chance to impact them and show that these things can work. And then as you've seen, we have this Ontario-wide program of network where patients get, 200 patients will get genomes, look at the PDOs as per Irene's high throughput screening. There'll be some epigenomics in there as well. We're thinking about ways to better integrate the data, molecular tumor boards to discuss the science, and hopefully these results will drive better decisions for the patients as the next step and also get them on more of our trials like ADOPT, which Irene has mentioned already. So this is exciting. I should mention this is now going to be taken over by my young colleague, Erica Zhang, to run this across Ontario. Yay, someone else. So we know that there are actionable findings on DNA today in pancreas cancer, and they matter and they should not be missed. You know, the KRAS wild types, the HRD status, do better on platinum, all these rare things. So if you add all that up, we're left with 73% that still are kind of back to the old chemotherapy. So putting on a more optimistic hat, how can we improve on that? Well, one thing is under HRD, we have noticed this tandem duplicator phenotype, which has been mentioned, which it is an HRD, but it responds to platinum much better. And this is from the COMPASS data. And we have an abstract going out now on that, which may be able to expand that grouping. The whole world is looking at immunotherapy to try to turn cold tumors hot. And while the first attempts at that in pancreas were failed, I think newer ideas will be better. And so stay tuned for that. All this RNA signature stuff, and Faiyaz is revising it on a monthly basis. But I really think that they're incredibly important. It might take some time to sort them out, but we need this because chemotherapy is not going away, and we have to get patients on their best chance to get the disease under control and respond while we're figuring out other things to do with them that might take some more time. So this will be impactful. And you've probably heard that KRAS has become druggable, and there's lots of new drugs coming along the pipeline that may influence the more common alleles in pancreas cancer, KRAS or PanCuRx ideas that may have a greater impact on patients than ever before. Although I don't think they'll work alone. I think they'll have to work in conjunction with other strategies and other drugs to try to get the best out of them. So I always end up with this slide because as a clinician, I have to reflect, how are we measuring success in real time with precision medicine and advanced pancreas cancer patients? I think we can agree that we can identify and reach a large population. We can get the tissue and we can study it, and we can look at the models and we can deliver information back to clinicians, which is no small feat. And we've worked really hard on that for the last decade. Building more and more options for them is something that has to keep happening so that we have places to move these patients to when we find them. And luckily, there's a lot more of these ideas today than there were even a few years ago. But the big checkbox is here. How do we measure patient efficacy, or how do we move that needle? We have to hit more patients than just the 13%. And that's what we're working on clearly, and it'll take a little bit more time. So my take-home message would be to any clinicians listening is support all these trials. Don't miss precision opportunities for any of these pancreas patients, because if you don't look, you won't find it. So my last slide, I would just like to acknowledge, I think the collaboration between us at Princess Margaret and the OICR has been synergistic. It really has led to some influential data, and the work has been done by so many people, some of them listed here and some in the pictures, which has been very gratifying for all of us. We couldn't have done it without the patient engagement, especially the patients and their families willing to take a chance with us and trust us on these studies. And of course, we couldn't do it without all the generous funders that are listed here. Thank you very much.